Thank you. Dr. Conaco and Dr. Ilag, for those wonderful lectures. The floor is now open to your questions. Yes, Divine.
My question is for Dr. Cecilia G. Conaco. First, Dr. Conaco, I would like to extend my congratulations and salute to your persistence to be still of service to the Filipinos despite of the fact that you were given the opportunity to be abroad, earn money there, but still for keeps, you decided to be here in the Philippines. That's a rare attitude of especially Filipinos we were given the opportunity to earn lots in the country where they have visited. My question, Dr. Conaco, is it is expressed in a portion of your PowerPoint that exposure to diversity must exposing diversity you must learn from nature solutions. Alam niyo po, when you talk to ordinary people, yung itong phrase na to, simple. Exposure to diversity to learn from nature solutions. Ang hirap ipaliwanag. Because I am a Tagalog writer. Pag sinusulat ko yung mga artikulo po, especially in science, napaka-technical. Bago ko matapos sa isang artikulo, I have to consult the English-Tagalog dictionary. And again, experimentuhin ko. Yung bang tin-translate ko, maintindihan o lalong nagulo yung isip ng mga nagbabasa. Kasi there are terms, especially mga technical terms na ang hirap i-Tagalog. That's why lalagay ko na lang siya as is, then later, bigyan ko ng konting paliwanag. But to you, paano niyo po ipapaliwanag itong phrase na ito, Dr. Conaco? Kasi po, talagang, kaya na pa ako nag-iisip, paano ko taturbahuin itong mga sinasabi po ninyo. Salamat po.
Yeah, thank you for that question. I think yung ibig ko pong sabihin sa statement na yun that we must explore diversity to learn from nature solutions is that kailangan nating i-recognize na most organisms or all organisms that have been living wherever in certain habitats all have experienced stress over millions of years. So kung ano yung itsura nila ngayon, that's a product of all of that history, right? So, hindi pa rin siya simple, pero so parang kayo, diba? You're shaped by your history, right? So you learn heartaches and stuff like that. It's the same thing, I think, with microbes and their hosts. So they learn from all of that, what shaped their current community now is all of the adversity that they face through evolutionary times, many millions of years. So ibig sabihin, every time we look at a particular sponge and its microbes or any life must be in a sponge, even your gut microbiome and you, it's probably been shaped through millions of years of evolution. And that partnership is important kasi na-maintain siya all throughout. So you can guess na kaya meron tong partnership na to because it's advantageous for the organism, diba? May benefit siya para sa inyo. So yung mga microbes na nasa sponge, they're important for the sponge to survive. Pag tinanggal mo yun or pag disturb mo sila, the sponge is going to be, not happy with that. It might not survive in the environment. But I'm talking about sponges in particular, but I think the concept holds true for all organisms that live in such close relationship with their microbes. So again, your relationships are shaped by all of these millions of years of evolution, and these particular relationships are really important for you to survive in your environment. Which is why we saw kanina na kapag disturbance sa partnership, the whole entire organism is sort of really negatively impacted. I guess that's all I meant to say. And that would only really scratch the surface, so we need to really look at a lot more different organisms to see ano ba yung possible combinations ng sponge at ng microbes that would also provide the ability to withstand the stress. So kasi hindi lang naman isang solution ang ginagawa ng nature. As you know, nature gives rise to such diversity. I could probably also stand here all day and talk about this. Pero yeah, so just to sum up, we need to look at all examples because all of these examples have been shaped over millions of years of evolution. So maraming combinations that are beneficial, and I think we need to know all of the different types. So we cannot just base all of our mga concepts on one type of partnership. Kasi marami pang ibang nag-work din na partnerships in nature. I hope that's all.
Question for Dr. Ilag? Yes, Dr. Dalisay.
Good morning po, Dr. Ilag. Congratulations on your talk. This is more of a comment rather than a question. So, I am very happy to inform you that the Philippines is advancing in mass spectrometry. And we will be getting the first multi-imaging mass spectrometry in the country. So, meron po tayo, advanced na po tayo sa when it comes to mass spectrometry. So the lab will be located at University of San Agustin, and it will be inaugurated by Secretary de la Peña on April 20. So first in the Philippines and third in the ASEAN region, magkakaroon po tayo ng multi-imaging mass spectrometry looking at different distribution of whatever substances and compounds na nakita niyo po kanina, looking at where are those BMAA makikita, for example, sa brain, things like that. So we could collaborate perhaps in the future on that aspect.
Yeah, so that's very exciting actually, but one precaution, so for example, there are a lot of mass spectrometers around and a lot of universities that have mass spectrometers. One difficulty is that mass spectrometry is a very dynamic field. So in the, let's say, first world countries, every 3 to 5 years, your mass spec is kind of old. But for me, I've worked with very old mass spectrometers. So I've worked with 20-year-old mass spectrometers. And so it's the work that I showed with BMAA were all on old instruments. So that's nothing to be scared about. But it's just that. But one thing is important is that when you get a new instrument, to invest on proper choice. But since you're already receiving it, and because it's not the first time that the Philippines receives these big grants or whatever to get mass spectrometers. So I hope, and I wish, I not just hope, I'm willing to, of course, be of help, and I'm interested to collaborate, of course. So.
Oh, that's very good for our collaborations. Any more questions from, yes, Dr. Dalmasio?
Morning. Thank you to our previous two speakers for the great talk. My question is for Dr. Conaco. So I would just like to ask if you have also explored the transcriptome of the microbes per se in the sponge, because while it is important to know the transcriptome profile of the sponge in response to environmental stress, it's also important to know the transcriptome profile of the microbes, because we want to know for the microbes who are remaining in the sponge, how are they helping or not helping the sponge cope with the stress in the environment?
Thank you for that question. The brief answer is no, we have not looked at the transcriptome of the bacteria yet, but that is definitely a direction to go. I think the only reason we haven't done it is technical and funding. But you know, it's something to look at. We haven't also looked at the metagenome of that community, the full metagenome. So marami pang questions that are open, and I hope people will be interested enough to look.
We can also open the questioning to the first two speakers, Dr. Rivas Reyes and Dr. Concepcion. Follow-up question? Okay.
I have a question also to Dr. Rivas Reyes. Sir, My question is, how do you know if CHIME is enough? Because CHIME is limited to the OTUs in the database. But sometimes you don't get the identities and you get OTUs as high as phyla, which is not really a relevant information if you just get phylum information. So if you get like very broad information after doing CHIME, so do you pick particular microorganisms and find their identities in other relevant databases?
Yeah, thanks for that question. So, methods question. So actually, Les, meron ng bagong database in DADA2, which is actually a little bit deeper and wider in terms of the database. So what that means is there's always progress in the methods, like Paul said. There's always new databases, and so it's important to sort of keep track of it. So yung data now, actually we use DADA2 now, and that can go hopefully from class order family down into genus level with the same sequencing depth and reads. So hopefully the databases are always increasing. And there's always limits, so there are always limits to the methods in the databases, and that's why at some point, and of course science is incremental, maybe we can analyze to the family level, and then later on we can use that same data and then reanalyze it, and then maybe we can go deeper, but sometimes we just have to wait until the technology catches up and the databases catch up. But that also tells you the importance of open access and having the data there so that other people can also analyze it. And I think this is a trend that I'm an advocate of, including all kinds of data, so that other people can analyze the data also when you do your statistical analysis. Because one of the issues we're now facing as scientists is replicability. A lot of people are asking, what's the real truth? Sabi mo, kape magaling. Sabi mo, hindi magaling. Alcohol. And we have to do all of these meta analysis of different studies. But if the data is there, then somebody can say, yeah, I can analyze the data myself using the latest statistical methods. So it's the same with the databases. So every time we publish, we have to submit the sequences. But beyond that, if we can archive our data, then we can come back to it too. And it's okay to say, I'm not sure. At this point, we can analyze only up to this level. We think this is what's happening, and that's fine. We can stop there. And it's important not to over-interpret the data also, right? So as scientists, we want to be able to link it to our hypothesis, but it's also important not to over-interpret and say this is also what we can do. Hopefully that's a good answer. I also want to touch base on yung paano explain ng scientific concepts, for example, in Tagalog. My experience is it always works, and this was really illustrated when Cecil talked, right? It always works when you think about analogies. Paano may explain sa lay people, yung analysis ni Cecil, it's about stress and humans, right? So, mas madaling maintindihan. And I don't know if you noticed it, but there was a buzz in the room when she started talking about stress in your life history, and love life maybe, or, she talked about that, and it's now becoming relatable. So, mas madaling ma-explain kung initially, not very deep, kasi yung analogy will always fail, right? And hindi mo nakaka-capture yung subtleties. Pero, if you start with, stress in the environment, it's like stress in humans. And that leads to resilience and resistance and all of these things. So, as an entry point for discussion, most people will be able to identify with that and will be able to think in those terms as an entry point. Obviously, maraming subtleties na hindi exactly pareho. The other thought that came to mind with that question is the question of mimicry, right? Like Cecil said, billions of years of evolution. And so, as an engineer, I always think about mimicry. How do I, nature has already, test proven a lot of these solutions over time. And so, how do I copy that? So, maraming examples in engineering. The most efficient pumps are shaped like, lotus leaves. The most efficient patterns are patterns found in nature. fractal designs, they're patterns found in nature. So many examples. Yung termite mounds, yung anay na punso, di ba? Those structures can be mimicked to have very efficient ventilation systems in buildings. Marami examples. And so, mimicry, in engineering, we always think about mimicking nature. Because again, we don't need to do all the prototyping if nature has already done it for billions of years among the universal solutions. Anyway, sorry, nakikisa ba ito sa question?
That's fine.
You give me the chance to
Yeah,
talk.
please. Okay.
I have so many questions in mind, but I'd like to address 2 points. Kasi baka hindi lang kami magkita ulit ni Professor Ilag. And then I can always talk to Dr. Conaco. I think you wrote me one time regarding my organism. I don't know. I think I saw your name one time. Yeah.
What was your organism?
The organism is Pyrodinium. It's a dinoflagellate. Yeah, but I was not able to really discuss this further with you. But anyway, I have some collaborators at the University of Uppsala. Yeah, Dr. Granelli. So I think now that I'm retired, I'm a professor emeritus already, I think I have to connect you to my former student, graduate student, Dr. Deo Onda, and then to the others at the Marine Science Institute, because you're working on the marine environment, on the marine organisms. And my former student is working on microbes, so I think you have to be connected with him. And then also Dr. Lilibeth can work with you on this. So I think you can work with them and train other people in the Philippines in the line of work that you're doing. So, pag-usap na lang ulit tayo. Gusto ko, ano na naman itong ating person from the media. Okay. There is the so-called Komisyon ng Wikang Pilipino. The head is a national artist, I forgot the family name, but he has been working with us. Itong tinatawag na transdisciplinary. Now we approach problems using both the natural sciences and the social sciences. In fact, the 2 international councils have fused, the International Council for Social Science and the International Council for Science. So, isa na lang yan. So, lagi nag-uusap yan. So, we now have a collaboration. We're coming up with a memorandum of agreement. The social scientists and the natural scientists. Itatagalog, ipipilipino nila ang mga gawa ng mga scientists. Okay? So, narinig niya akong magsalita tungkol sa red tide, yung head ng komisyon. So, yun ang unang project. So, marami pang iba. So, pwede kang tulungan siguro nila. Okay.
Maraming salamat, President. Ako'y substitute for the commission AKR. So I think it's amazing that the gathering of minds, and we have a media champion here and mine. So any more questions?
Yes, may I ask you a question? About your card, can you talk about it also? Because several times in problem also.
And you happen to deal with the marginal sectors of our society. How do you really try your best to commune with them and explain fully yung essential ng inyong mga dinidiscuss, which are most of the time highly technical po?
Alam po ninyo sa aming research, kami po'y kailangan humingi ng permiso sa mga communities, fishing communities. Required po yun in the Wildlife Act, which is a law. So meron po kami sabi prior informed consent from the community surrounding the biodiversity where we collect samples. So meron kaming primer, meron kaming meeting sa barangay, pwedeng Pilipino, Taglish, Oman, Ilonggo. Tapos, we work with the fisher folk. Kasi you'd be surprised na meron talagang alam yung mga fisher folk na hindi natin alam. Yung isa pang research ko, nauukol naman doon sa mollusk na mas alam natin, yung conoidean mollusk, yung conus, pero kami, inaaral na rin namin yung mga pinsan na mga conus na tawag ay turid. So, mas malalim ang kanilang atirahan. But anyway, meron kaming paraan para kuhanin yung mga ganong klaseng samples dahil yung mga fishermen naandun sa Mactan at Bohol, they cater to tourists, Japanese tourists, who like to get these tiny shells. So meron silang mga old nets na kinukumpul-kumpul nila at iniiwan nila doon sa dagat, certain depth. Tapos biglang ecologically, nagagalit together yung mga mollusks. Ngayon, meron pang ibang paraan na naglalagay ng bait para makakuha na ang good bait para makuha ng mga kanilo. Yan po nasa knowledge ng mga communities, ng mga fisher folk. So, I think it's important to engage them by showing that they are part of the research, they are part of the value chain, and they are the stewards of the biodiversity. Kaya, pag kami mangyari na talagang nagkaroon ng parang drug prospect, meron tayong agreement with them na sila ay merong share. So, I think that's an incentive for them. But, on the other hand, tama yung sinasabi ni Cecilia, sabi ni Ma'am Rod, na kailangan gawing analogy or allegory yung mga ating mga research natin. Pero sa totohanan, hindi man lang yung analogy yung sa sponge. I used to start my talks before saying that the sponge is my ancestor, so I bow to the sponge. Because the sponge is also something I studied for many, many, many years. Ang dami-daming compounds dyan. You know, yung transcriptome na pinakita ni Cecilia doon na nagbabago yung response. Yung mga proteins, yung mga molecules na nagre-respond, yung mga genes na nagre-respond on encoding for the proteins, they're very similar to the human genes and proteins, cell-cell adhesion, cell-cell adhesion na pa yung mga iba, yung mga immune complement activation. Can you imagine? Ang sponge mayroong complement activation. Parang tao. So, totoo lang. Bakit maraming anti-cancer sponges compounds? Kasi nga, yung sponges, maraming regulatory mechanism similar to humans. Okay? To control cell proliferation. So, mga ganong mga examples, magandang to educate the public. Kasi, totoo lang, yung scientific literacy sa bansa, medyo may resistance na dito.
Nakatapusan magpakanin natin sa third. Can we hear from the educators from the other institutions? Pagbigyan muna natin. Dr. Pérez, please.
Good morning, Franco Teves from Mindanao State University, Ilag Institute of Technology. I'd like to address this question to Dr. Conaco. Simulation experiments are very important, especially for environmental studies and probably for the survival of species in the future. But I would like to ask if the simulations that you have used are based on model, and what do you think could be other factors that should be included there, because there are only two, I think, temperature and pH, but changes in dissolved gases, for example, as a result of increased temperature, and then plus the melting of the ice caps, about salinity, will it not also impact on the interaction between the sponges and the microorganisms that are present?
Thank you for that question, because I love that people are sort of opening up the other questions that, my lab can't do everything. So definitely, I think our study is limited to just acidification and temperature, because that's what's covered in the project, in the program. Pero definitely there's so many other stressors that our marine ecosystems face, like eutrophication, there's of course low DO, salinity, reduced salinity is also something. All of these things I think also need to be explored, but we can't sort of put all the stressors all together kasi parang complicated na i-interpret yung mangyayari sa data. But just looking at them perhaps one by one or in combination, a few in combination, I think is already really very useful. So you can see how things are responding, and you can see which organisms are more resistant or less resistant. So little by little, I think we can sort of tick all of the questions one by one. Pero you raise a very important question that there are so many other stressors that are faced, and that we need to also address these, not just just pH changes and temperature, although these are also really global kasi yung impact.
Can I just have one question for Dr. Ilag? I'd like him to share what he did with
Fraction with laser desorption ionization. So that maybe sounds very technical, but essentially, instead of doing chromatography or other, let me say that again. So instead of doing the traditional multi-step way of analysis, we basically have developed a strategy where you have the solid phase extraction, that is when you capture the analytes, to be on the same substrate that you actually directly put into the mass spectrometer, like a multi-mass spectrometer, which is a laser desorption-based mass spectrometer. So you could immediately analyze it. So in simple terms, it means that if your workflow would take, for example, 30 minutes just to analyze because you have to do chromatography and mass spectrometry, this one could do it in seconds because it's based on laser desorption. Okay. So the story goes that we patented that out of a kind of like exercise because we thought, oh, this is an interesting idea, and the university was interested in encouraging people to protect their intellectual property. But in Sweden, the intellectual property belongs to the inventor 100%. So it's not, the university has no say on whether we want to patent it or not. But we did, and it was a journey that was fun, but took many years, and it took us to know the different funding agencies, etc. But at some stage, there were no more funding for actually patenting things. So then I had to resort to asking people, my mommy, etc., to help also. But at the same time, you know, but in the end, it was just getting so difficult that in the end, there was, you're actually make a company in some sense because you have to have an entity, a legal entity that will protect the patent and continue to generate income to protect the patent, etc. But to cut the story short, we then created a company, and now it was last year funded by the university took a percentage of the company, so funded it a bit, and we have already, but we already had value by then because we already managed to have an EU patent and a US patent covering the technology. And now actually, and it's good you asked the question was raised because I didn't have time to say that I would look for collaborations, of course, with people, and my accountant who's owning less than 10% of the company with me, so I own the majority. He's collaborating with India to actually exploit the patent. But then I was thinking that I should also maybe, I mean, I know the person that he's collaborating with in India, but I thought it would be good to have that collaboration in the Philippines as well. So, but I have not had as much contact, and so this forum actually helps me to do that. And to think that I'm doing it now in the time of me being here because of the company. So I'm not using company time from the university. So, because we're very strict in Sweden that you're not supposed to use your time in the university. And the lab, for example, in the university, I can't use for if I do any contribution to the company, even for basic research. For example, if I do a basic aspect and I could rationalize its basic research, but there is a link that will help the company, then it's also considered of commercial value. And then I'm not supposed to. I know I'm a bit more obedient than other people. I know some people work around that, but I take days off when I actually have to work with companies.
Thank you, Dr. Ilag. I hope some of our young scientists will be inspired to be entrepreneurs too. So, some more questions? Yes, Dr. Sadar.
Last question siguro to. I just noticed that most of the studies done so far are focusing mostly on the bacteria only. Being a mycologist, actually I have been recommending this also sana that they can also include, in particular for example, the sponges. We also did work on the fungal population, and we noticed that we wanted us to separate also the causal residents and the indigenous population. And in fact, I would like to ask the Dr. Conaco here if they also did try to separate those are the actual residents of the bacteria in their sponges and the so-called causal residents who might be transients only, because this has an implication in the overall dynamics of the population. Thank you.
Yeah, so for sponges, well, the short answer is no, we did not separate, but we do compare with water samples to see if there's any overlap. But I think a lot of sponge work has already been done to really show that what you see in the sponge when you sequence it is surprisingly really specific to the sponge, and the water community, which might be, just added transiently when it's filtered in, this is sort of parang, it's diluted out, that effect is diluted out. So what you really see most is the...
Thank you for all your questions. I have to bow to our last president and we'll give her
Sorry,
the last
na-excite ako sa
question, last
sinabi
comment.
ni yung dalawa. I'd like to mention what my former student, Deo, did in connection with my organism. Tawag ko Mai kasi medyo nauna akong nagtrabaho doon. The one causing red tide in the Philippines, Pyrodinium bahamense. Okay, so what he did, he studied the extracellular, meaning the endosymbionts and the exobionts. And then there are changes in the exobionts and the endobionts at the different life stages. So it shows a lot, the result shows a lot about the ecosystem, how the organism also adjusts to the ecosystem. So hanggang ngayon, pinagtatrabahuan pa niya yan. So I think after his PhD in France, when he came back, sabi ko, lumabas ka, mag-aral ka, doon naman ng iba. So now that he's here, he's doing a lot. So including pollution, micropollution, nanopollution, microorganisms and so on. It's the reason why I said I think we need to do a lot of work on this. So microorganisms, how they can do something to help in pollution even in the marine environment. By the way, meron na isolate, kay kwento ko na, no? May na-isolate sa Manila Bay, isang malaking dinoflagellate. Gusto daw ipangalan sa akin kasi yung isang estudyante ko nasa Tokyo. I think pagiging successful siya doon. Kasi that particular dinoflagellate can eat a lot of the pollutants in the area. So there's hope there's some hope, I hope, I think. So it's a big dinoflagellate feeding on other dinoflagellates and other microorganisms. And my other student working at the University of Tokyo has this paper already being reviewed, and I think it's reviewed already, and it's going to be named after me. I hope it passes. So, talagang interesado ako sa pollution. Thank you.
Dr. Concepcion cannot resist herself, so
I have a question For Dr. De Los Reyes.
Oh, okay.
So we have bacteria that are specialists. They degrade cellulose and they also consume hydrogen sulfide. Meron ka bang gustong explore na uses for bioremediation or waste management and all that? Because we have them in the lab.
So thank you for the question. Kasi meron din akong answer dun sa isang question. Sorry. So a big issue is cellulolytics because a lot of solid waste, for example, right? Landfills. In fact, we have research and papers in landfill microbiology. And in fact, in Hawaii, they now have these hot landfills, elevated yung temperatures landfills, and it's both metabolic as well as siguro may volcanic activity din. But in those areas, maybe merong sulfolobus or other organisms that are cellulose degrading, as well as sulfide oxidizing. So that's one issue. When you think about waste, a lot of it is cellulolytic, including for biofuels. We have enzymes, cheap cellulases. We can get biomass. When I say biomass now, I mean agricultural biomass, and convert that to biofuels more easily. Because one of the most expensive things are the cellulases conversion. Your first hydrolysis step. Once it's solubilized, the whole thing fermentation is a little bit easier. So yes, I think there are ways of thinking about it. There's a big company that's based in Sweden, I think, or no, Denmark, in Novozymes, right? They're all about enzymes and they're all about microorganisms. And so this is a big potential for commercialization potential. I had a quick, and I didn't have time to talk about all of the things that we do, but one of the things that we study fundamentally is what we call microbial community assembly processes, and it's a whole field. So, saan galing yung mga microorganisms? Yun yung tanong, right? And it can be, and how do they assemble? It can be deterministic, in other words, they evolve and they respond to stresses and environmental factors, and that's what shapes the microbiome. Or they can come from other processes that are more stochastic. In other words, major random depending on what's happening. So if I operate 2 reactors at the same time, I control everything. Environment, pH, temperature, everything. When I look at the microbiomes, they're not going to be exactly the same. In other words, there's something a little bit random about the process. So this is called stochastic. And now there are ways of modeling these things and figuring out how much percentage is stochastic and how much is deterministic. And in fact, in another talk tomorrow, I'll talk about some of that. How do you mathematically approach that so that you can say X% is stochastic and X% is deterministic? In the anaerobic digesters I had shown earlier, the data, it's mostly deterministic. But there are other systems, depending on the function, that are more stochastic. So this is really basic microbial ecological theory. But once we understand some of that, like you said, we can think about causality, which is another topic that I'm really interested in, which is the talk of my topic of my talk tomorrow. Afternoon at UP Diliman is causality. So there are lots of interesting questions, and there are ways now of simulating and mathematically modeling these things. And that's the beauty of thinking about it in a different way. You can make it a little bit more quantitative, and yung naman yung engineering hat. So there are all of these advances that are really, really exciting. Sorry, I can't resist to say, we're